These scientists were jointly awarded the Nobel Prize in Medicine or Physiology 39 years after they published their research. Let's talk about it. In 1992, Edmond Fischer and Edwin Krebs, not the Krebs of Krebs cycle, although he was also a recipient of the Nobel Prize. Let's start this all over again. In 1992, Edmond Fischer and Edwin Krebs were recognized for their groundbreaking research concerning reversible protein phosphorylation as a biological regulatory mechanism, which is just a fancy way of saying controlling and regulating cellular activity by adding or removing phosphate groups to proteins. Welcome to episode 2 of Spotlighting Nobel Prize winning research in medicine or physiology, explaining it to you so that you understand, telling you its relevance and how it has shaped modern medicine as we know it. Now, to fully understand their work, there are some things we have to get out of the way. The first being the fact that you have to ditch the way you think about proteins as meat, fish, or milk and adopt them for what they truly are: molecular machines responsible for running almost every aspect of cellular function. Think of proteins as enzymes, antibodies, receptors, and hormones, etc. Proteins are the final product of your genes and they do nearly everything. The second thing to get out of the way is that cells communicate, and by communication I mean they send and receive signals. And by signals I mean things like hormones, drugs, neurotransmitters, temperature changes, etc. Cells can also regulate themselves and others. That's how your cells know when to divide, when to die, and when to produce or destroy something that is no longer needed. This response to signals actually has a fancy name called signal transduction. That was a lot of talking. Now that we've laid the groundwork, let's get to the meat of the matter. Before the 1950s, scientists knew that cells responded to signals, but they didn't know exactly how those signals triggered a response inside a cell, or how cells could so precisely regulate those responses. The dominant theory was if a protein was needed to elicit a response or regulate the activities of a cell, the cell made it. If the protein wasn't needed, the cell broke it down. And honestly, I see how that could make sense. It's like turning a factory on or off by building or destroying machines when needed. This was to say that proteins worked when they were made and stopped working when they were destroyed. While that may be partly true, it wasn't exactly the case and didn't explain how cells could so fastly respond to things like adrenaline or insulin. Then came Fischer and Krebs. They were studying how an enzyme that breaks down stored sugar called glycogen phosphorylase could switch between its active form, which is phosphorylase A, and its inactive form, which is phosphorylase B. They not only discovered that the active one had a phosphate group and the inactive one didn't, but that they could also switch between active and inactive by the removal or addition of phosphate, respectively. The thing is, these enzymes were previously thought to be two separate enzymes, not one enzyme that could switch from one form to another. Fischer and Krebs' discovery meant that the activities of proteins could be regulated by adding, reducing, or removing phosphate groups. And if you can regulate the activities of proteins by reversibly adding phosphates, then you can regulate cellular activity and function. You know how I made an analogy of what scientists thought about how proteins were regulated before Fischer and Krebs, right? How if you needed something done in the cell, you either built or destroyed the machines, i.e. the proteins? Fischer and Krebs showed us that you did not have to necessarily destroy anything. All you had to do was switch proteins on or off by adding or removing phosphates. They had uncovered a fundamental mechanism for how cells responded to signals and regulate themselves. Today, we know that reversible phosphorylation is involved in so much, from cellular growth to immune response to learning to memory and cell death. More than 60 years later, their work is all over modern medicine. Because disruptive phosphorylation is linked to diseases like cancer, diabetes, Alzheimer's, etc. Entire classes of drugs like inhibitors of an enzyme called kinase, which is actually the enzyme that does the phosphorylation, are based on manipulating these phosphorylation pathways. Fischer and Krebs discovered the cell's molecular switch, and we've been using it to fight disease ever since. As I was reading about their work, I just kept on wondering, these guys received a Nobel Prize 39 years after their research was published. That's almost 4 decades. But then I thought, this packs for a motivational punch. Then I thought of it this way. Their work was not born for that era. It was built for the future. And it only became apparent as biology advanced. So if you have what you think is an ingenious idea or invention, and people around you just don't seem to understand the value or just how much potential you see in it, just remember this. Your work or idea may be ahead of its time. Welcome to my science pep talk. Hope you learned a thing or two.